Biology, a self-teaching guide by Stephen D. Garber. Chapter 2, Cell Structure. Microscopes. Anything as small as a cell was unknown before the sophisticated optics became available. During the 17th century, ground lenses that were being used for eyeglasses were first arranged at opposite ends of a tube, creating a small telescope. It was a sort of step to the invention of the microscope, one of the first constructed by the Dutchman Anton van Leeuwenhoek. With the light microscope, the examination of specimens was facilitated by thinly slicing them, allowing light to pass through. By strands by straining the specimen, it was possible to emphasize internal structures. For instance, staining cellular fluid pink and staining solid, hard structures purple provided increased contrast, enabling those who study cells, known as cytologists, to discern these structures more clearly. While some light microscopes permit researchers to view objects as much as 1,500 times their actual size, stereomicroscopes was also called dissecting microscopes, magnified objects from only 4 times to 80 times. With two eye specimens, the advantage to this low-powered three-dimensional view is that researchers can investigate much larger objects such as the venation of insect wings. Since the invention of light microscopes, the most significant technological advance for cell researchers was the development of electron microscopes, which occurred in the early 1930s. It has not only improved the ability to see smaller structures with greater magnification so they appear larger, but also enhanced the ability to see things more clearly or with additional resolution. When it was discovered that the illumination of specimens with blue light under the light microscope lens considerably greater resolution than with any other color of longer wavelength, researchers speculated that using shorter wavelengths might add even more resolution. However, wavelengths shorter than those of violet lights are not visible to the human eye. This problem led to the invention of the transmission electron microscope, which utilizes a beam of electrons that travels in short wavelengths than those of photons in visible light. These electrons are passed through a thinly sliced specimen within a vacuum to prevent any electrons from being deflated and absorbed by the gas molecules in the air. Then the electrons are focused with electromagnetics on a photographic plate, producing an image that is considerably better than that obtained with the light microscope. Then in the 1950s, the scanning electron microscope was invented, which focused electrons that bounced off the specimen. Since the SEM has less resolving power than the TEM, it doesn't require a vacuum and allows researchers to view some smaller organisms alive. The most recent advances in microscope technology allow scientists to observe living cells with even greater magnification. One of the newly developed techniques is called contact X-ray microscopy. Another microscope, the scanning tunneling microscope, enables scientists to photograph molecules. Biology, a self teaching guide by Stephen D. Garber. Chapter 2. Cell structure. Cells. With the availability of the first microscopes, researchers began to observe the microscopic structure of many substances, and in 1665, the Englishman Robert Hooke described having seen what he called cells in a part of a cork. He used this term because the cork appeared to be composed of thousands of tiny chambers that resembled the individual sleeping rooms in monasteries at the time, which were called cells. He was not aware that he was viewing just the cell walls, which were the only structures remaining from what had once been living cells. Hooke's initial discovery led to other advances, such as the finding that unlike plant cells, which have thick cell walls, animal cells lack such a wall and instead have only a thinner, generally more flexible plasma membrane. Cells were then found to exist independently or as one small part of an organism consisting of many cells, a multicellular organism. Hooke was the first to discover that some organisms consist entirely of a single cell. The unicellular organisms, such as thousands of species of bacteria and protozoa, carry out all necessary life-supporting functions within one cell without the help of other cells. In contrast, multicellular organisms have cells with specific functions, and together, the aggregate of cell embodies a complex organism. Biology, a self-teaching guide by Stephen D. Garber. Chapter 2. Cell structure. Cell theory. It took about 150 years after Hooke discovered cells before several important related facts were articulated. Two German scientists, Matthias Schleiden and Theodor Schwann, were the first to explain, in 1838 and in 1839, respectively, the basic tenets of what we now call cell theory. 1. Cells are the fundamental unit of life. 2. Cells are the smallest entities that can be called living. And 3. All organisms are made up of one or more cells. Biology, a self-teaching guide by Stephen D. Garber. Chapter 2. Cell structure. Cell structure and cell size. The longest cell are certain nerve cells, neurons, which can reach over a meter in length. While an ostrich egg is 1,500 times the size of a human egg cell, which is 14 times the size of a human red blood cell, itself as much as 35 times the size of many small single-celled microorganisms, most cells do have one thing in common. They tend to be quite small. While the size range reflects considerable diversity, most cells are 0.5 to 40 microns in diameter. Small cell size is thought to be a function of the restriction placed on cells by the ratio of surface area to volume. Cells are constantly absorbing molecules from the surrounding medium and releasing molecules into the surrounding medium. These processes are more readily accomplished when a cell is small and the ratio of surface area to volume is quite large. As a cell increases in size, the amount of volume inside the cell increases much more rapidly than the amount of surface surrounding the cell, and in time, the cell becomes too large to maintain a stable internal environment. Many scientists believe that it is more difficult for the nuclear material to maintain control over the entire internal environment when a cell is over a certain size. Therefore, if a small nucleus is most often the rule, then the upper limit is placed on the size of most cells. Biology, a self-teaching guide by Stephen D. Garber. Chapter 2, Cell Structure. Cytoplasm and nucleoplasm. Except for the nucleus or nuclei, everything within the plasma membrane is called the cytoplasm. The nucleoplasm consists of the contents within the nuclear membrane. The cell's interior is composed largely of a complex solution as well as a heterogeneous colloid. A solution is a homogeneous mixture of two or more components in which the particles of the different substance are so small that they cannot be distinguished. A colloid usually contains particles that are too small to be seen but large enough not to form a true solution. The particles don't settle out at an appreciable rate. Different areas inside a cell may be in different colloidal states. Some areas are a sole state. That is, the colloidal particles, which are usually macromolecules, are randomly dispersed throughout the area. The other parts of the cell may be in a gel state, in which the colloidal particles interact and form a spongy network. A colloid in the gel state forms a semi-solid. Changes from the soil to the gel state may be stimulated by change in pressure, agitation, temperature, pH, salt concentration, or the concentration of other substances. Some mixtures containing proteins and lipids have properties of true solutions as well as some sol gel capabilities of a colloid, and this often typifies the internal fluid environment of a cell. Biology, a self-teaching guide by Stephen D. Garber. Chapter 2, Cell Structure. Cell membrane. The cell membrane is the membrane surrounding the cytoplasm at the cell's surface. The membranes contain varying amounts of proteins and lipids. The specific types of lipids, which are fat-soluble substances located in the cell membrane, are primarily phospholipids and steroids. The total thickness of the cell membrane, which is sometimes also called the plasma membrane, is only about 80 angstroms. Biology, a self-teaching guide by Steve D. Garber. Chapter 2, Cell Structure. Movement through the cell membrane. In addition to providing protection, shape, and strength to the cell, the membrane helps regulate the flow of material in and out of the cell. Many processes work in concert to maintain the cell's constant internal environment. The condition of constant internal environment is referred to as chemical homeostasis, and the method a cell employs to maintain this constant environment was described below. The two basic methods require different energy inputs. When the movement of molecules occur on its own, without any organized energy input from the cells, the movement is termed passive transport. When the cells expend energy to move molecules from one location to another, the process is known as active transport. Diffusion. One type of passive transport called diffusion may occur in a dynamic system such as a gas or liquid, in which particles may be moving in a manner that appears random. In a system where diffusion occurs, a net movement of particles can be observed over time from regions of higher concentration of that substance to regions of lower concentrations of that substance. The particles moving from the area of greater concentration will produce a concentration gradient ranging from the point of highest concentration to the points farthest away, where the lowest concentration occurs. The particles don't move in a straight line, but the concentration gradient is instead a result of many small movements. The tendency is for molecules to move down a concentration gradient. That is, they go from an area of highest concentration of the molecules to where the concentration is less. The movement leads to a point where molecules are eventually dispersed evenly throughout the environment, where they are all able to move equally freely. The movement of the particles can produce at different rates depending on such factors as the concentration of temperature of the medium. Some membranes are impermeable to the movement of anything through them. Other membranes may exclude some substances, but they are permeable to others. The partial or differentially permeable membranes is called semi-permeable membrane. Osmosis. Another example of passive transport is osmosis, which involves the movement of water through a semi-permeable membrane. The pressure exerted by dissolved particles in a solution or a colloid known as osmotic pressure moves water across a permeable membrane. When a semi-permeable membrane separates two solutions of pure water, the two sides are at equilibrium. That is, the osmotic pressure is equal on both sides. When the osmotic pressure is the same on both sides of a semi-permeable membrane, each solution is said to be osmotic or isotonic. This means that each medium has the same concentration of osmotically active particles and each has the same osmotic pressure, so neither tends to lose or gain appreciable quantities of water via osmosis. Another alternative exists when a cell is in a medium with a higher concentration of osmotically active, dissolved, or colloidal particles, rendering a higher osmotic pressure. In such a situation, water moves across the cell membrane into the medium that is hyperosmotic, also called hypertonic, relative to the solution inside the cell. When a cell is in a medium with a lower concentration of osmotically active, dissolved, or colloidal particles, the solution thus has a lower osmotic pressure than that inside of the cell. The cell is said to be in a medium that is hypoosmotic or hypotonic relative to it. So water moves into the cell. To avoid confusion, one should be careful to note the point of reference of these terms. For instance, when the internal environment of a cell has a higher concentration of osmotically active particles than the external medium, this means that the medium inside the cell is hyperosmotic relative to the external environment. Either way one chooses to state it, the water still moves from hypoosmotic environment into a hyperosmotic environment. Water moves from where the osmotic pressure is low to where the osmotic pressure is high, to where there are more osmotically active particles. If this process is allowed to continue, all things being equal, the solution on either side of the semi permeable membrane should eventually become osmotic to each other. If particulate matter is poured into one of the two previous isomatic solutions, the particles dissolve into solutions. Then the equilibrium will be upset and the osmotic pressure that now exists between the two sides will be unequal. Water will move from the area of lower concentration of dissolved molecules. The permeability of different types of plasma membrane varies significantly. An amoeba's plasma membrane has less than 1% of the permeability to water of the membrane of a human red blood cell. Most plasma membranes are quite permeable to water and other substances such as many simple sugars, amino acids, and lipid soluble substances. Plasma membranes are relatively impermeable to larger complex molecules such as polysaccharides and proteins. Permeability to small molecules indicate there are pores through which these particles can pass. It may be that these pores open and close according to the cell's needs at a given time. The membrane's permeability to lipids substances is related to the lipids that make up part of the membrane. Lipids are able to dissolve right through the membrane. In cell membrane, in the cell membrane are carrier molecules that help move certain substances in and out of the cell. They are described in the following discussion on facilitated diffusion and active transport. Facilitated diffusion. One of the two main types of transport that involve active as opposed to passive transport is known as facilitated diffusion. Here, carrier molecules move substances either in or out of a cell in a manner that increases that rate of diffusion across the membrane. The mechanism involved combines molecules that are to be moved across the membrane with the carrier molecules in the membrane. In such a manner, molecules are picked up on one side of the membrane and released on the other side. A common example of facilitated diffusion is the rate at which the cells take up glucose, an important simple sugar in many biological systems. Most cells absorb glucose much more rapidly than the rate at which such absorption would occur without any added help. In concert with the carrier molecules, as the cell started to use the glucose rapidly, the concentration on the inside of the cell decreased, and because of the increased concentration gradient, glucose is moved into the cell more rapidly than usual. Facilitated diffusion works in both directions. Sometimes it is equally important to move substance into the cell as it is to move other substances out of the cell. Certain hormones can affect the rate at which some substances move across cell membrane. For instance, insulin increases the rate of glucose uptake by some tissues. While glucose normally moves along concentrated gradient, facilitated diffusion helps increase that rate. Active transport. To move substances such as glucose across the cell membrane against the concentration gradient, energy has to be expended. The process by the substance move against their concentration gradient is termed active. There are types of active transport systems. Some take up amino acids, others move peptides, nucleosides, or potassium. These substances are moved with an appropriate carrier system. Both of the most extensive studied active transport systems use the high energy molecules adenosine triphosphate ATP. In some cases, it appears that specific globular proteins within the cell membrane temporarily change shape under certain conditions. The modification may temporarily open a pore wide enough for the transported molecules to pass through. When active transport is involved, energy from ATP may help change the conformation of the carrier molecules so it will pick up the molecules that is to be moved, or ATP may help detach the transported molecules from its carrier once it has been moved to the other side of the membrane. Movement of large particles across membranes. The plasma membrane has the capacity to engulf small particles on either side of the cell and then fuse with these particles, or sometimes the portion of plasma membranes that surrounded the small particles simply separate, pinching itself off and drifting away. When liquid or macromolecules are engulfed by the membrane and are then moved to the other side, the process is known as pinocysis. The same process, but on a larger scale, that takes on larger particles, are phagocytis. These engulf particles, pinch off with parts of the membranes, and move in or out of the cell. The process that moves particles into the cell is endocytosis, and the process that moves particles out of the cell is exocytosis. Biology, a self-teaching guide by Stephen D. Garber. Chapter 2, Cell Structure. Plant cell wall. All cells have a plasma membrane, but some also produce a non-living outer cell wall. The cell wall is a production of the cytoplasm that supports and provides shape for the cells. Most bacterial cells have a rigid or semi-rigid cell wall outside the plasma membrane that is usually composed of a polymer glucose derivative attached to amino acids. Sometimes certain bacteria have an additional layer outside this known as lipopolysaccharides, a polymer composed of lipid and sugar monomers. The complex polysaccharide that composes most plant cell walls is cellulose, a related compound. The cellulose wall allows a wide range of compounds to pass through easily. It is the cell membrane that determines which material may enter or leave the cell. In complex multicellular plants, the primary cell wall is the outer layer that is laid down first by the young cells. And in some of these plants, where two cell walls come in contact, another layer is laid down between the primary cell walls. This is known as the central or middle lamella, which is one continuous layer between the cells, and it is usually composed of pectin, a complex polysaccharide, generally in the form of calcium pectate. It is the pectin that binds the cells together. Plant cells that become harder or more woody usually do so by adding layers to the cell wall known as the secondary wall. This wall is also deposited by the cytoplasm and is inside the primary wall. Rather than being laid down in a loose network as in the primary cell wall, the long thread-like structures that make up much of the cell wall, known as cellulose fibrils, are laid down parallel to one another in the secondary cell wall in a series of layers or lamellae. Each layer has fibrils oriented at angles of about 60 degrees to the fibrils of the next lamellae. The parallel fibrils, the number of layers, their orientation, the total thickness, as well as added substances such as lignin, help strengthen the cell walls. Biology, a self-teaching guide by Stephen D. Garber. Chapter 2, Cell Structure. Plasmodesmata, and gap junctions. Plasma membrane connection, known as desmosomes, are thin cytoplasmic connections that often run from one plant cell to the next through pores in the cell walls. These pores are called plasmodesmata. Facilitates intercellular exchange of materials such as sugars and amino acids. Gap junctions, sometimes called nexus junctions, are areas of low electrical resistance between animal cells across the electrical impulses small ions and molecules may pass. They seem to function similarly to the plasmodesmata that connect plant cells. When animal cells are in water with a low relative osmotic pressure, water will move from the area of low osmotic pressure into the cell with higher osmotic pressure, and they will ultimately burst due to the cell detention or turgor. Plant cells can usually withstand the swelling without bursting because their sturdy cell walls counterbalance the low external pressure. Biology, a self-teaching guide by Stephen D. Garber. Chapter 2, Cellular Structure. Cells' internal structures. Nucleus. Relatively large, distinct membrane-enclosed structures of several kinds are found in most kinds of cells, though not all. The notable exceptions are bacteria and cyanobacteria, often referred to interchangeably as blue-green bacteria or blue-green algae. The largest, most well-defined subcellular structure or organelle is the nucleus, unless even larger vacuoles are present. Vacuoles are large, fluid-filled spaces found in many cells, particularly plant cells. The nucleus contains the long, thin structures composed of deoxyribonucleic acid, DNA, and protein, the chromosomes. These structures contain genes, which are the individual units of information that inform the cells what to do and how and when to do it. All the instructions concerning the life process of the cells emanate from the chromosomes. Nucleic acids are a major group of organic compounds that are composed of subunits called nucleotides, which contain carbon, hydrogen, oxygen, phosphorus, and nitrogen. It is the sequence of the nucleotides that gives the specific nucleic acid its distinctive properties. Dio, dio, is it D? Yeah, deoxyribonucleic acid and ribonucleic acid (RNA) are two important nucleic acids found in cells. They are fundamental in the storage of transmission of genetic information that controls the cell's function and interactions. A dark area within the a dark area within the nucleus of most cells, seen when the cells are not dividing, is known as nucleus. The area This area contains the high concentration of ribonucleic acid, RNA, and protein. Sometimes a nucleus contains several nucleoli, which are formed by a specific region on the chromosomes called the nuclear let me see nucleolar organizer. All the nuclear structures are embedded in a viscous colloidal material, the nucleoplasm. The nucleus is bonded by a membrane, the nuclear membrane, which consists of two complete membranes similar in structure to the cell membrane. The nuclear membrane is very selective, restricting certain substances that readily pass through the cell membrane into cytoplasm, but are unable to pass through the nuclear membrane. The nuclear membrane is perforated by many large pores that facilitate the passage of substances in and out of the nucleus. Attached to the outside of the nuclear membrane are many thin membranes of endoplasmic reticulum, or ER, that appear to be important in assisting the two-way flow of materials to and from the nucleus. Endoplasmic reticulum. The plasmic reticulum, ER, is a series of double-layered membranes found throughout the cytoplasm that provide surface area for enzymes to catalyze chemical reactions, particularly those leading to protein synthesis. The ER forms a multi-branched channel between the outsides of the cell and the nucleus, connecting to the cell membrane and the nuclear membrane, thus expediting the export of cellular materials. Endoplasmic reticulum may be present with or without the outer surface lined with small particles called ribosomes, which are the actual sites of protein synthesis. Endoplasmic reticulum without ribosomes is known as smooth endoplasmic reticulum, and this is where some steroid hormones and other lipids are synthesized. The endoplasmic reticulum that is lined with ribosomes is referred to as rough endoplasmic reticulum. These terms, smooth and rough endoplasmic reticulum, relate to the structure's appearance in electron micrographs. Ribosomes. Ribosomes are tiny granules that can exist either in the cytoplasm as free-floating organelles or along the outer surface of the endoplasmic reticulum. Cells contain three main types of ribonucleic acid, RNA, each synthesized from DNA. The RNA found in ribosomes that functions in protein synthesis is known as rRNA, which stands for ribosomal RNA. Ribosomes contain high concentration of rRNA and protein. In addition to existing singly and attached to endoplasmic reticulum, some ribosomes occur in clumps or clusters, which are collectively known as polyribosomes or polysomes. Golgi apparatus. The Golgi apparatus, or Golgi body, is an organelle that looks like a stacked, flattened, smooth, oval bowl shaped membrane. It is through that certain chemicals that are synthesized in the ribosomes move along the endoplasmic reticulum to the Golgi apparatus, where they are concentrated and stored until released by secretory vessels produced by the outer portion of Golgi apparatus. In addition to functioning as an area where substances such as proteins pass through and are stored before being secreted, the Golgi apparatus is also involved in processing some proteins and carbohydrates, synthesizing them into more complex molecules by combining them into glycoproteins and mucopolysaccharides, some of which are secreted in the form of mucus. Lysosomes. A lysosome is a membrane enclosed storage vessel. The membrane is composed of a single layer, unlike the double layered membrane in the nucleus. Lysosomes appear to arise from Golgi bodies. They contain strong digestive hydrolytic enzymes that act as the cell's digestive system, making it possible for the cells to process within its cytoplasm material, such as the particles taken in by pinocytosis and phagocytosis, defined earlier in the chapter under the heading Movement of Large Particles Across Membranes. Lysosomes also break down old parts of the cell into the organic molecules from which they were constructed. These molecules are then reused by the cell. The importance of lysosomes has been established in studies concerning the metamorphosis of frog larvae. When tadpoles' tails shrink in size, their constituents are being broken down by lysosomes, and their organic molecules are then reabsorbed to fuel the growth of new structures such as frogs' budding arms and legs. Mitochondria. Often referred to as the powerhouse of the cells, mitochondria supply the cells with chemical energy in the form of the high energy molecule adenosine triphosphate, ATP. Slightly larger than the lysosomes, these double membrane organelles are often largest of the organelles in the cytoplasm, although plastids, vacuoles, described later, and the nucleus are also quite large in many cases. Because to supply cells with energy, mitochondria are most abundant in cells with high energy requirements, as in the growing root tip of a plant or in an animal's muscle cell. Each liver cell contains as many as 2,500 mitochondria. The outer membrane separates the mitochondria from the cytoplasm, and the inner membrane has many inward directed folds or cristae that extend into a semi-fluid matrix. It is along the cristae and in the matrix where the energy transfer enzyme system that manufactures ATP are found. ATP is used in cell division, in muscle cell contraction, and as an energy source for many other cellular activities throughout living organisms. When more sugar is available in a cell than it requires, fat droplets found next to many mitochondria will produce fat. Then, when an energy deficit arises, the mitochondria will break down the fat droplets to yield energy. Excess fat droplets are transferred to specialized fat storage cells. Plastids. Like mitochondria, plastids contain DNA, RNA, and ribosomes and are able to replicate themselves. Plastids are rather large cytoplasmic organelles that can be seen with a light microscope. They occur in most photosynthetic plant cells and also in photosynthetic protists, single celled organisms such as some algae. Plastids are composed of two membrane layers. The outer membrane lacks folds and encloses the organelles, regulating the movement of material between the cytoplasm and the organelle's interior. The interior membrane folds throughout the internal proteinaceous matrix known as the stroma. These internal membranes occur in sheets called lamellae, which often form stacks or grana. These stacks are connected by a single lamella. Different types of plastids have been reorganized and each appear to develop from a common structure or something or sometimes from another. Those that are white and colorless and that store oils and protein granules as well as energy rich starch formed from simple sugars are the leucoplasts. Plastids filled with starch are called amyloplasts. Plastids containing yellow, orange, or red pigment are called chromoplasts. These are found in flowers and fruits. Chloroplasts are the chromoplasts that, in addition to yellow, orange, or red pigment, also contain green pigment or chlorophyll, which uses energy from the sunlight to manufacture organic molecules such as sugar from inorganic molecules. In addition to chlorophyll, some of the other pigments, including carotenoids and xanthophylls, remain in temperate zone leaves in the autumn after the green pigment has broken down. It is these pigments that account for the autumn color. Vacuoles. Vacuoles are membrane bound, fluid filled spaces in both plants and animal cells, although they are most prominent in some plant cells. Much of the total volume of most plant cells is composed of a single vacuole surrounded by its membrane, the tonoplast. The liquid inside the cell, the plant vacuoles, is called cell sap, which is mostly water with some dissolved substances. Cell sap is usually hyperosmotic relative to the external medium surrounding it. So, in times of need, when available, water will move in through the vacuole's membrane via osmosis, keeping the cells turgid. Vacuoles often contain a high concentration of soluble organic compounds such as sugar, amino acids, some proteins, and several pigments. Many plant species store toxic chemicals in the vacuoles that do not harm the cells. If an animal eats the plant, however, the contents of the ruptured vacuoles can have an adverse effect on the animal. Different kinds of vacuoles have different functions. Some single celled organisms have contractile vacuoles, which are important in expelling excess water and waste from the cell. Many cells have vacuoles or vesicles that are formed when the cell membrane takes in small particles by phagocytosis or pinocytosis, which may then be digested by enzymes from the lysosomes. Biology, a self teaching guide by Stephen D. Garber. Chapter 2, Cell Structure. Microfilaments. Within a cell's cytoplasm are many long protein fibers arranged in microfilaments that are only about 6 nanometers in diameter. Microfilaments function in the maintenance of cell structures and movement. Depending on the local conditions in the cell, microfilaments can exist either as individual subunits or together in strings of subunits. Microfilaments are believed to be important components of the contractile filaments that exist in some cells. They may be associated with cytoplasmic steaming, a constant movement of cytoplasm in the cell that is evident in many plant cells under the light microscope. The microfilaments may also be involved in a cell movement, and when in more complex forms, they may be associated with the contraction of some muscle cells. Biology, a self-teaching guide by Stephen D. Garber. Chapter 2, Cell Structures. Microtubules. Also in the cytoplasm are microtubules, which differ from microfilaments in many respects. The outer diameter of each microtubules is considerably larger than that of a microfilament, ranging from 20 to 25 nanometers. 1 million nanometers equals 1 millimeter. 1 billion nanometers equals 1 meter. Furthermore, microtubules are hollow and vary in length from less than 200 nanometers to 25,000 nanometers. Microtubules also differ from microfilaments in terms of their chemical makeup. Sometimes microtubules act as tracks along which the cell's organelles move. Microtubules orient themselves towards each pole of a cell, and chromosomes move along them during the cell division. A distinctive internal microtubular arrangement also determines the arrangement of a cell's cilia, flagella, and sperm tails, which are discussed below. Cilia and flagella. Cilia and flagella, singular, cilium and flagellum, are long, thin organelles that project from the surface of some cells and have the capacity to beat back and forth or in a corkscrew fashion, moving the cell along fluid or moving fluid past a stationary cell. They appear on some protists, on certain lower plants, and on some animals. Many eukaryotic organisms have flagellated sperm cells. Eukaryotic and prokaryotic bacterial flagella differ structurally and mechanically. The difference between cilia and flagella is mostly a matter of length, though not diameter, and sometimes they are different according to the number. Many short ones together are called cilia, while an isolated long one is usually a flagellum. The function of these organelles is primarily locomotory, to propel or pull a cell through liquid. Some protists also use cilia to move particles into their gullet, a part of the cell membrane that is similar to a mouth. Multicellular organisms commonly have cellulated cells lined internal passageways. Their constant motion helps move materials through tubes and ducts and preferred direction. In the preferred direction. In all types of cells except bacteria and blue algae, cilia and flagella are cytoplasmic outgrowths from the surface of cells surrounded by extensions of the plasma membrane. At the base of the long cilium or flagellum is the basal body, which supports and gives rise to the rest of the long stalk. It consists of 9 triplets of microtubules arranged in a circle. The basal body may be where the protein subunits assemble to form the microtubules that extend through the rest of the long structures, which are themselves composed of a circle of 9 pairs of microtubes with another pair of their center. The 9 plus 2 arrangement is characteristic of all cellular cilia and flagella except those found among bacteria and blue algae. Centrioles. Basal bodies and centrioles are structurally identical, although the basal bodies are found at the base of cilia and flagella of all cells, while centrioles are found only in animal cells, generally located near the nucleus. Centrioles occur in pairs. Like basal bodies, they are composed of 9 sets of triplets, all arranged lengthwise, creating a cylindrical structure. Not all cells contain centrioles, but most animal cells have them, and they are always composed of the same typical pattern, a cylinder formed by 9 sets of triplets that, when cut in a transverse section or cross section, looks like a pinwheel. Besides playing a role in the formation of microtubules, centrioles become active during cell reproduction. During interphase, both pairs of centrioles are located just outside the nucleus, arranged at right angles to each other. As mitosis or meiosis begin, they move apart and organize the mitotic apparatus known as the spindle, which is composed of microtubules extending from the pole to pole through the cell's interior. It is along these microtubules that the chromosomes align at metaphase, then moving apart during the remaining stages of cell division. Toward the end of cell division, during telophase, the centrioles replicate, so each daughter cell in turn has 2 pairs of centrioles located just outside the nucleus. For more comprehensive discussion of cell division, see Chapter 3. Eukaryotic and prokaryotic. The differences between cells of bacteria and blue algae and those of most other organisms are customarily cited to distinguish between 2 distinct cellular types, eukaryotic cells and prokaryotic cells. Eukaryotic cells. Most of the preceding parts of this chapter concerns eukaryotic cells. Eukaryotes are those plants and animals with eukaryotic cells. These include all the protists, fungi, plants, and animals. Their cells have a nucleus enclosed within a nuclear membrane, inside of which are located the chromosomes, the long filamentous structures along which occurs the units of inheritance called genes. Those chromosomes are composed of nucleic acid and proteins. Eukaryotic cells also contain mitochondria, Golgi bodies, or Golgi apparatus, endoplasmic rectum, and most of the other organelles so far described. When present in these cells, chlorophyll is located in chloroplast. And if these cells have cilia or flagella, they always possess the previously described 9 plus 2 structure. All the content of eukaryotic cells are enclosed with a plasma membrane, and in some cases, as with many plant cells, the plasma membrane is also enclosed within a cell wall. Prokaryotic cells. Prokaryotic cells are thought to be more primitive than eukaryotic cells. Bacteria and blue algae are prokaryotes, which is to say their cells are prokaryotic. These cells do not have a nuclear membrane. Instead, they have a nuclear area known as nucleoid that in most cases contain single cellular chromosomes. In prokaryotic cells, the chromosomes are composed entirely of nucleic acid without protein. Their ribosomes are also structurally distinct from those in eukaryotic cells. When flagella-like structures are present, they don't have the 9 plus 2 fibular structure. The prokaryotic cells contain no mitochondria, no Golgi apparatus, no lysosomes, and no vacuoles. In addition, prokaryotic cells have cell walls containing muramic acid and characteristics not found among eukaryotic cells. In many prokaryotic cells appears an enfolding of the plasma membrane known as a mesosome, which may be involved in the production of new cell wall material following cell division, and which may be associated with the replication of DNA during cell division. Mesosomes, the mesosomes may also help break down food molecules to supply the cells with energy. Most photosynthetic prokaryotes contain photosynthetic membranes and chlorophyll, but these materials aren't enclosed in chloroplast.